Hey everyone, Dr. D here, and in this video we are going to be doing an Exam 2 review for Biology 1406. This will cover chapters 4, 5, and 6. So let's go ahead and get started. Yes, Wicket. Hey, you guys want to see Wicket? He's come to say hi, you guys. It's Wicket. Sweet Wicket. Hi, Wicket. Come help me do the exam review today. All right, let's get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
All right, everyone, welcome back. Sorry about my voice. I have a bad cold right now, so this may be a little bit shorter and less edited than my usual videos because I'm trying not to cough as I talk, and I'll edit out any coughing. So let's go ahead and begin with Chapter 4. Again, as always, please study all your lecture handouts. Study every slide because I can't remember everything for the exam review, and what I try to do for the exam reviews is, I make sure to mention the more important things, but that doesn't mean I mention everything in an exam review. So please, just to be safe, study all your handouts before you do the exam. And but let's go ahead and get started with Chapter 4. Remember, this covered carbon. We talked about organic chemistry. You should know that organic chemicals have at least one carbon and one hydrogen. You should know that carbon can make four valence it has four valence electrons, which means it can form four covalent bonds with a variety of atoms. Remember that? So it can make four bonds. It always makes four bonds. Remember that when carbon only makes single bonds with its neighbors, it has a tetrahedral shape, a pyramid shape. But when it forms double bonds, it becomes planar. Remember that? The bonds become planar. Okay. Remember, we talked about different types of ways that carbon molecules can differ: length, double bond position, branching, presence of rings. Importantly, we talked about the shorthand for organic chemistry. You see here the shorthand. Here you can see shorthand. Remember, hydrocarbons are organic molecules consisting only of carbon and hydrogen. Next, it's important that you remember your isomers, structural isomers versus enantiomers. Remember, enantiomers are isomers that are left and right isomers. They are mirror images. They are mirror images of one another. Whereas cis trans isomers, this is where, the isomers appear on the same side, or these extensions appear on opposite sides from one another. That makes the difference between a cis isomer and a trans isomer. So please know your different types of isomers for the exam. Next, we talked about functional groups. This was a very important part of the chapter. You should remember all of your functional groups from the video. So just to do a quick review here, this is what I need you to know about each of the functional groups. You should know, obviously, what it looks like. For example, a hydroxyl group is a O and an H bonded to your carbon backbone. It can also be written as HO. A hydroxyl group makes a molecule an alcohol. So you should know that. You should know that a hydroxyl group is polar. So those are the things to know about hydroxyl group. They are polar. They form alcohols, and they are written OH or HO. For carbonyl groups, again, this is when you have an O double bond to your carbon backbone. Now, remember that there are two types of carbonyl groups, ketone and aldehyde carbonyl groups, and I'll show you the difference between the two. Remember, when you have an O double bond to a C, if that C has a C on both sides, that is a ketone carbonyl group. However, if you have an O double bond to a C, and that C does not have a C on each side, for example, there's an H on this side, that is an aldehyde carbonyl group. So you should know the difference between a ketone and an aldehyde carbonyl group. And you should also know that this is a polar group. This is a polar functional group as well. Next, we talked about the carboxyl functional group. This is when you have an O double bond to a C as well as an OH bonded to that same C. You should know what it looks like here. You should know that this forms an acid. So molecules that have carboxyl groups are acids. And you should know that they exist as the non-ionized form on the left, or the unionized form on the left, and the ionized form on the right. Remember, it can be written as a negative charge like this. So this is a carboxyl group in water. This is a carboxyl group outside of water. Next, we have the amine group, or amino group, I should say, the amino group. This is a N with two H's attached to your carbon backbone. Amines have this. And you need to know that this is a basic functional group. You should remember that this is a basic functional group, which means outside of solution, the functional group is NH2. However, in solution, it becomes NH3+. And this is because it sequesters a proton from the solution. Right, so you should know, again, amino groups are NH2 or NH3+. They are basic functional groups. Next, we learned about the sulfhydryl group, or SH, also written as HS if it's on the left side of the molecule. These are thiols. And remember, the important thing I wanted you to know about SH, or sulfhydryl groups, is that when two of them come together, they form a disulfide bond, a proper covalent bond. That was the important thing we touched on. So please remember that two sulfhydryl groups, when they come into contact, they form a covalent bond called a disulfide bond. That was the important thing to remember about these. Next, we talked about the phosphate group, and this is a phosphate group. It has negative charge. Phosphate groups have negative negative charge. And so you should know that this is a phosphate group of phosphorus with 4 oxygens. And lastly, we talked about the methyl group, or the methyl group, which is CH3. And what you need to know about the methyl group is that it is the only non-polar group out of the 7 we talked about. Those are the important things to know about the functional groups. All right, and that takes us to the end of chapter 4. I think we touched on all the important parts here for chapter 4, so that's good. Let's go ahead and take a break time. I'm going to take a throat lozenge and rest my voice for a little bit, and then I'll be back after a nice break time with Gizmo and Wicket. We'll carry on with chapter 5. All right, welcome back from break time with Gizmo and Wicket. Let's go ahead and carry on with chapter 5, the structure and function of large biological molecules. You should know, for this chapter, you should know that there are 4 classes of biomolecules: the carbohydrates, the lipids, the proteins, and the nucleic acids. But remember, only 3 of them are macromolecules. The one that's not a macromolecule are the lipids, right? The lipids are not macromolecules. You should know that dehydration reaction or dehydration synthesis is how 2 monomers are bonded together with the loss of a water molecule to form a covalent bond, whereas hydrolysis is how these polymers are disassembled and broken back down by using water to break the bond. You should know about carbohydrates. Carbohydrates are made up of monosaccharides, which can be, multiples of CH2O. So if you see a molecule that's made up of CH2O or some multiple like C6H12O6, that's a monosaccharide. So you should know that glucose is C6H12O6. And then remember that you can have different length sugars as well, different length 3 carbon sugars, 5 carbon sugars, 6 carbon sugars. But I'm not going to ask you to know all the different isomers, like the difference between ribose and ribulose, or glucose, galactose, and fructose. Okay, you should know that sugars can form ring structures in water. You should know that the bond that forms between monomers when you link, or between monosaccharides, is called glycosidic bond or glycosidic linkage. You should know that the glycosidic linkage is the bond between monosaccharides. See, here and here. Definitely know that. You should know polysaccharides. We talked about different polysaccharides, and I'll let you know what we need to remember. With starch, you should know that starch and glycogen are a type of polysaccharide for storage of energy. You should know that starch is made by plants and it's less branched. You should know that glycogen is made by animals and it is much more branched. You should know, on the other hand, cellulose is made by plants and it's not for storage of energy. It is for structural. It's for the cell wall. It's for structural purposes. And you see, look, it looks flat, right? The cellulose is flat and it forms hydrogen bonds. And here's something important to remember. This is important to remember. You should know that alpha glucose isomer is used to make starch and glycogen, while beta glucose isomer is used in the making of cellulose. You should also know that, why can us humans digest starch and glycogen, but we cannot digest cellulose? Remember, it's because we do not have the correct stomach enzymes to do so. You should know that chitin makes up the cell walls of different fungi, as well as the exoskeleton of insects. Okay, moving on to lipids. You should know that lipids are usually mainly hydrocarbons, and they mix poorly, if at all, with water. And then we talked about fats, phospholipids, and steroids. So please know that a fat is made up of a glycerol attached to 3 fatty acid tails. Remember that? A glycerol and 3 of these fatty acid tails makes up a fat. And do you remember that there is an ester linkage in the, connecting the tails to the glycerol is an ester linkage right here. And you should know the difference between a saturated fat and an unsaturated fat. This one is this one here is a saturated fat, is it not? Because there's only single bonds in the tails, right? There's only single bonds in the tails, so it's a saturated fat. Unsaturated fats, you should know they have a double bond in the tail. These are the things I need you to know for the exam, for the difference between saturated fats and unsaturated fats. In a saturated fat, you have only single bonds in the fatty acid tails. In a saturated fat, they tend to be solids at room temperature. In a saturated fat, they tend to be from animal products. And lastly, this is probably the trickiest one. In a saturated fat, you have more hydrogens in the fatty acid tails because they're only making single bonds. You can have more hydrogens. Let's look at an unsaturated fat now. Okay. An unsaturated fat has at least one double bond in a fatty acid tail. An unsaturated fat tends to be liquid at room temperature, like oil. An unsaturated fat is from plant or fish products. And an unsaturated fat has fewer hydrogens than a similar saturated fat. All right. Next, we talked about phospholipids, and you should know the structure of a phospholipid. You should know that a phospholipid has a glycerol head, a phosphate group which is negatively charged, as well as a choline, and you have two fatty acid tails. So this is the structure of a phospholipid. And you should realize that this part here called the head is the hydrophilic head because of these charges. The hydrophilic head mixes with water. Now the tails here are known as hydrophobic tails because they don't mix with water. They're made up of carbon and hydrogen only. And you should know that phospholipids make up the membranes of your cells by forming a phospholipid bilayer. Next, you should know about steroids. Steroid hormones are lipids, and they have 4 fused rings, such as cholesterol. See, you have 4 fused rings. On the exam, you should know that when you see 4 fused rings like this is a steroid hormone, a type of lipid. All right, next, we talked about proteins. You should know that proteins have all different kinds of functions. Proteins are made up of amino acids. You should know that proteins are made up of amino acids. Amino acids link together to form peptide bonds. And when you link amino acids together, you form polypeptides and proteins. This is amino acid. Amino acids have remember, you should know that amino acids have an amino group. You should know that amino acids have a carboxyl group. You should know that amino acids have an alpha carbon. And then amino acids have an R group, right? All amino acids have an R group. Here are the 20 amino acids. I may give you an amino acid and ask you if it's a non-polar amino acid, a polar amino acid, an acidic amino acid, or a basic amino acid. How would you tell the difference? Well, if the R group only has carbons and hydrogens, it's going to be a non-polar R group or non-polar amino acid. If the R group has hydroxyl groups or sulfhydryl groups, then it's going to be a polar amino acid. If the R group has a carboxyl group or a negative charge, then it's an acidic amino acid. If the R group has a positive charge, it's a basic amino acid. And again, you should know that when amino acids link together, they form peptide bonds. You should definitely know peptide bonds form between amino acids. You should know that all proteins, all polypeptides have an amino end or N terminus with an amino group hanging off, and all proteins or polypeptides have a carboxyl end or the C terminus with a carboxyl functional group as well. You should know that the backbone links together while the side chains point out the side. You should know the difference between primary structure, which is simply the sequence of amino acids in the protein, the secondary structure, which is made up of alpha helices and beta sheets, the tertiary structure, which is the final folded protein, where the protein folds into its final configuration, and this is thanks to the interactions between the R groups. Remember, there were 4 different R group interactions that help with tertiary structure. I'm going to touch on those in just a second. And then remember, most proteins never form a quaternary structure, but some do. The quaternary structure results when a protein has to work with other proteins. It has to work in a functional unit with other proteins as well. It can't do its job on its own. So again, primary structure is the list of amino acids in a protein. Secondary structure includes alpha helices and beta sheets. Tertiary structure is the final folded protein. And remember, the reason tertiary structure forms are those R group interactions, including hydrogen bonds, ionic bonds, hydrophobic interactions, and disulfide bridges. See, hydrogen bonds, ionic bonds, disulfide bridges, and hydrophobic interactions. You should know that sickle cell, we talked about how sickle cell is formed, right? I may ask you, how is sickle cell formed and what is responsible for sickle cell? Remember, there is a amino acid substitution here at the 6th amino acid residue from glutamic acid to valine, valine being a nonpolar amino acid. This mutation causes the beta subunit of hemoglobin to misfold with an exposed hydrophobic region. And when this forms a quaternary structure with other hemoglobin proteins, this exposed hydrophobic region causes quaternary structures to link together, forming filaments that stretch out the cell. If that's, if you need a refresher on that, don't forget you can watch the chapter 5 video. You should know that environmental and chemical conditions such as pH, salt concentration, temperature, these types of things can cause a protein to denature. That means lose its fold. And once a protein denatures, you should know it does not typically renature. Okay, once you denature a protein, it will not renature. And lastly, we talked about the nucleic acids. You should know that the nucleic acids are, they consist of DNA and RNA. Okay, DNA, deoxyribonucleic acid, RNA, ribonucleic acid. You should know that these nucleic acids are made up of monomers called nucleotides. Each nucleotide consists of a nitrogenous base, a pentose sugar, and a phosphate group. Remember, there are some, the pyrimidines, which include C's, U's, and T's, and the purines, which include A's and G's. Remember, in DNA, the sugar is deoxyribose, whereas in RNA, the sugar is ribose. So again, all nucleotides, all nucleotides have a phosphate group. All nucleotides have a pentose sugar, though the pentose sugar of DNA is deoxyribose, while the pentose sugar of RNA is ribose. You should know that the nitrogenous base can be different, right? This was what makes up the code of DNA and RNA. You should know that the nitrogenous bases of DNA include C's, T's, A's, and G's, whereas the nitrogenous bases of RNA include C's, U's, A's, and G's. You should know that the pyrimidines are C's, U's, and T's. Remember, one pyramid can cut. Pyrimidines are C's, U's, and T's, whereas the purines are A's and G's. Okay, so know which ones are the purines, know which ones are the pyrimidines for the exam. And then remember that every time you form a strand of DNA or a strand of RNA, you should know that the bonds that form are called phosphodiester bonds. You should know that phosphodiester bonds form when nucleotides are linked together. Again, just to review, when monosaccharides are linked together, that was called a glycosidic bond. When amino acids were linked together, that was called a peptide bond. When nucleotides are linked together, that is called a phosphodiester bond. And remember, different lipids do not link together. Different lipids do not link together. This is why they are not known as macromolecules. And remember, this is a strand of DNA or RNA, and you should know that every strand of DNA or RNA has a 5' end with a phosphate group and a 3' end with a free hydroxyl group. Next, you should know that the two strands of DNA are anti-parallel. Anti-parallel, which means when you look at the two strands of DNA, one strand is going 5' to 3' in one direction, while its complementary strand is going the opposite direction, 3 to 5. So the two strands of DNA are going in opposite directions. Another thing you should know is that about complementary base pairing. You should know that on one strand of DNA, G pairs with C on the complement strand. And you should know that A's pair with T's. And in RNA, you should know that A's pair with U's, but C's still pair with G's. Okay? And one last thing, remember, the backbone of DNA consists of sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Okay? These were the important things to know about Chapter 5. So I hope this helps. Let's take another quick little break time with Gizmo and Wicket. Let me go rest my voice a little bit, come back with Chapter 6, and hopefully we'll be prepared for the exam. All right, everyone, welcome back from break time with Gizmo and Wicket. Let's start with Chapter 6, the last chapter for Exam 2, A Tour of the Cell. Let's see, you should know the basic features of all cells. So I might ask you, like, which of the following is not a basic feature of all cells? And these are the basic features of all cells. You should know that prokaryotic cells, they do not have a nucleus or membrane-bound organelles, but they do possess a nucleoid. Okay. They have cytoplasm, nucleoid, they have ribosomes, they have plasma membrane. You should know that eukaryotic cells, they have the membrane-bound organelles, including the main one, the nucleus, which is a double membrane of the nuclear envelope. Okay, we should know some of the parts of the eukaryotic cells. You should know that animal cells have, a nucleus. We have the nucleolus, all of these parts here, membrane-bound organelles that we go through. There's also centrioles. Remember, the centrioles are made up of microtubules. We learned that later in this chapter. You should know the parts of the nucleus, right? These are the things you should know. The nuclear envelope is a double membrane. There are nuclear pores in the nuclear envelope. The nucleolus is where the rRNA is synthesized. Your DNA is made up of chromatin, which is DNA wrapped around histone proteins. You should know that the nuclear lamina is made up of intermediate filaments, right? Remember that from the end of the chapter. The nuclear lamina is made up of intermediate filaments, and it supports the shape of the nucleus. We should know about ribosomes. Ribosomes synthesize proteins. Ribosomes are either free in the cytosol or bound to the ER or nuclear envelope. Ribosomes have a large subunit and a small subunit. And remember, this is important. Ribosomes are made up part of protein and partly of RNA. Remember the endomembrane system? The nuclear envelope is the only membrane that's continuous with another membrane, and that's the endoplasmic reticulum. You should know that these two are the only two that are continuous, which means connected to one another. The rest are connected only by vesicles. You should know the difference between the smooth ER and the rough ER. Remember the functions of the smooth ER. You need to know these are the functions of the smooth ER, because I might ask you what is not a function of the smooth ER. And you should know the functions of the rough ER. Remember to synthesize proteins and then to send them, distribute them via transport vesicles. You should know the function of the Golgi. Remember, it's the shipping and processing center of the cell. You should know that the Golgi is made up of flattened membranous sacs called cisternae. You should know that the cis face of the Golgi accepts proteins from the rough ER, and then those proteins are modified, and the trans face of the Golgi ships the proteins away. You should know that lysosomes are known as digestive organelles. They contain hydrolytic enzymes. You should know what phagocytosis means. You should know what autophagy means. And then we talked about various types of vacuoles or large vesicles. Food vacuoles, contractile vacuoles, and central vacuoles of plants. Next, we talked about the mitochondria and the chloroplasts. You should know, first of all, you should know the endosymbiont theory. Remember that it suggests that the early ancestor of eukaryotes engulfed in oxygen using non-photosynthetic prokaryotic cell, which became the mitochondria, and then later on also engulfed a photosynthetic cyanobacteria, which later became the chloroplast. Okay, so again, the endosymbiont theory is it explains the origins of the mitochondria and the chloroplasts. The mitochondria and the chloroplast then became, organelles inside of our cells, but they were originally bacteria. And by the way, these are the evidence. This is the evidence. So if I might ask you, what is the evidence that supports the endosymbiont theory? And this is the evidence right here. Next, we wanted to know the structure of the mitochondria. Remember, the mitochondria has a smooth outer membrane and then a convoluted inner membrane. The folds of the inner membrane are called cristae. So you have the outer membrane, the inner membrane. The space between the two membranes is called the intermembrane space, and the fluid in the center is called the matrix. You should also know that mitochondria contain their own circularized DNA, as well as their own 70S ribosomes. You should know that the mitochondria is the site of cellular respiration, where ATP is made. Next, we talked about the chloroplasts. You should know that the chloroplasts are found in plant cells. Chloroplasts are found in plant cells as well as algae. You should know that there's the outer membrane, the inner membrane, and then these stacks of membrane called granum. Each each pancake in the stack is called thylakoid. The fluid outside the stack is called stroma. The fluid inside these stacks is called the thylakoid space. And the membrane of these stacks is called the thylakoid membrane. You should also know that these chloroplasts contain their own circularized DNA, as well as 70S ribosomes as well. Next, we talked about the peroxisomes. You should know that the peroxisomes, these contain enzymes to remove hydrogen atoms from, fats. They work in the breakdown of fats. Okay. But yeah, and they can also form hydrogen peroxide. So again, the functions of the peroxisomes use oxygen to break fatty acids into smaller molecules, so breakdown of fats. All right. Next thing, we learned about the cytoskeleton. You should know of the different fibers of the cytoskeleton: microtubules, microfilaments, intermediate filaments. You should know that microtubules serve as monorails for vesicular transport, for these vesicles to move along. You should know that microtubules are the thickest of the three components. Microfilaments are the thinnest, and they're made up of actin proteins. And the intermediate filaments are in between in diameter. You should know that the microtubules are constructed of proteins called tubulin, right? Dimers of alpha and beta tubulin. You should know that the microtubules form the centrosome, right? Which is made up of the centrioles. See, the centrioles make up the centrosome, and that plays a role in separation of DNA during cell division. You should know that microtubules are responsible for, or at least necessary for the flagella and the cilia to function. You should know that flagella work by beating back and forth, whereas cilia work with a power stroke. You should know that the flagella has a basal body that anchors the flagella to the cell membrane, and as well as dynein, which are walking motor feet to help with the bending of the flagella. And remember the 9+2 arrangement here: 9 doublet microtubules surrounding 2 singlet microtubules. All right, so you should know that microtubules are required for the flagella and the cilia. Next, we talked about microfilaments, which, remember, you should know these are the thinnest of the cytoskeletal components. They are made up of actin. They form the cortex of the plasma membrane. You should know that microfilaments are important to form the microvilli of your intestinal cells. You should know that microfilaments are important for muscle contraction. They are important in cytoplasmic streaming and pseudopod motility. And lastly, we learned about the intermediate filaments as well. You should know that intermediate filaments do not do any dynamic processes. They are static. They're there for static support. And remember, intermediate filaments, they also play a role in the nuclear lamina support of the nucleus, and they help to fix different organelles in place. You should know about the cell wall of plants. Plants form a cell wall of cellulose. You should know about the ECM, or extracellular matrix, of animal cells, and you should know it's made up of collagen, proteoglycan, and fibronectin on the outside, and these link to integrin anchor proteins. You should know that the transmembrane anchor proteins are called integrin. Integrin links to fibronectin and collagen, as well as proteoglycan. And on the inside, integrin links to microfilaments, but you should know that integrins are the anchor proteins of the ECM of animal cells. And then lastly, we learned about the cell junctions. You should know that plasmodesmata are channels connecting plant cells. You should know that tight junctions are these are proteins that press together neighboring cells, preventing leakage of fluid. You should know that desmosomes fasten cells together into strong sheets. And you should know that gap junctions provide cytoplasmic channels between adjacent cells. And these three are found in animal cells. All right. Well, thank you for bearing with me in this this review. I'm sorry I couldn't be more animated as usual. I'm very feeling under the weather right now. I think I'll go take a nap. So again, please study all your slides, study all your notes to do really well on the exam. You can do it. I know you can. Study hard. Don't just memorize. Remember, try to understand all the concepts. Work hard. Try to understand all these concepts, and I'm sure you'll do fine. Don't forget the full-length videos are there for you to watch as well if anything on the review seemed confusing. All right, I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.